to our channel Bioholic with your Friday educator Olivia. It is the part 8 of the photosynthesis, and after the C3 cycle today we will learn about the C4 and CAM pathway. Okay, so not wasting more time, let's start. Previously, we have learned about the Kelvin cycle or C3 cycle, where we have seen that the Rubisco is fixing the carbon dioxide and ultimately glucose is produced. Last Friday, we also learned about another thing, that is photorespiration, which occurs in the high environmental temperature and high oxygen concentration inside the cell. And in this process, we have seen that RUBP combines with oxygen and not with carbon dioxide. And here phosphoglycolic acid and phosphoglyceric acid both are formed as the first stable product. And in the mitochondria, when glycine is converted into serine, carbon dioxide molecule is released from the cell. And also another ATP is used in this process, but there glucose cannot be produced. So here no glucose is formed, ATP is used, thus there is also waste of energy and also there is loss of carbon atoms. That's why photorespiration is also called as a wasteful process. And to prevent this, some plants are evolved. Those are called as the C4 plants. The C4 photosynthesis is an adaptation of the C3 pathway that overcomes the limitation of the photorespiration, improving photosynthetic efficiency and minimizing the water loss in hot environments. And this C4 cycle occurs in approximately 1,500 species of 19 families of angiosperms, but most of the plants are monocotyledons, which belongs to Gramineae and Cyperaceae, like sugarcane, maize, sorghum, oat, bajra, etc. And also there is some dicotyledons with C4 cycle. Those are Euphorbia, Amaranthus, Chenopodium, etc. In these C4 plants, some special characteristics are seen. Here, some adaptation of leaves are found, and this type of anatomy is named as Cran's anatomy. Here, green bundle sheath cells are present around the vascular bundles, that means the xylem and phloem, and these bundle sheath cells acts as protective layer against the gaseous exchange for both oxygen and carbon dioxide. Thus, it is preventing oxygen to combine with RUBP. Hence, photorespiration cannot take place. And also here is dimorphic chloroplasts present in leaf cells. Chloroplast of bundle sheath cells or Cran cells are larger and without grana, and mesophyll chloroplasts are small and with grana. This is the structure of the leaf anatomy of C4 maize. Okay. Now the C4 pathway, that is also known as the Hatch-Slack pathway. It owes the names to the discovery by Marshall Davidson Hatch and Charles Roger Slack. And as I said before, that it is an evolved mechanism of carbon dioxide fixation. We know that to make glucose, carbon dioxide is necessary for the Kelvin cycle. But as the bundle sheath is preventing gaseous exchange, then how carbon dioxide will enter the Kelvin cycle? Let's see. As the carbon dioxide cannot enter as gaseous form, that's why in the cytoplasm of mesophyll cells, carbon dioxide combines with water to form carbonic acid. As carbon dioxide cannot enter the cell as gaseous form, that's why at first in the cytoplasm of mesophyll cells, carbon dioxide combines with water to form carbonic acid. An enzyme called phosphoenol pyruvate carboxylase adds the carbonic acid to a 3-carbon molecule called as phosphoenol pyruvate or PEP. And then they both are combined and ultimately form a 4-carbon molecule called as oxaloacetate or oxaloacetic acid, which enters the mesophyll chloroplast. As in this pathway, the first stable compound is the 4-carbon molecule oxaloacetate. That's why this pathway is called as C4 pathway. Okay. Nextly, malate dehydrogenase reduces the oxaloacetate into malate using NADPH produced from the light reaction. Then, malate is exported from the mesophyll cells through plasmodesmata, arriving in a bundle sheath cells. The malate enters the bundle sheath chloroplast. Then, NADP-dependent malic enzyme breaks down malate into pyruvate and carbon dioxide while reducing NADP plus into NADPH. So, thus, carbon dioxide enters the bundle sheath cells. And here, Rubisco is present, which incorporates the carbon dioxide into C3 cycle. And ultimately, as we know the C3 cycle, the glucose is formed. Again, here I will explain the full process. As the bundle sheath cell acting as a wall and preventing the entrance of the oxygen or carbon dioxide, thus oxygen cannot enter the cell. But we need carbon dioxide to produce glucose. And they are very clever. That's why they make a chemical compound, that is the carbonic acid, which is formed due to the combination of carbon dioxide and water. And the phosphoenol pyruvate combines with the carbon dioxide and makes oxaloacetate. As this oxaloacetate has 4 carbon, that's why this cycle is also called as C4 cycle. Then the oxaloacetate is reduced in malate, which is then transferred to bundle sheath cell from the mesophyll cells. And in the bundle sheath cells, by the help of the NADP+, the malate is cleaved into pyruvate and carbon dioxide. The carbon dioxide enters the Kelvin cycle and thus glucose is produced. And on the other hand, the pyruvate again enters the mesophyll cells, and here by the help of one ATP molecule, again it is regenerating the phosphoenol pyruvate for the next cycle. So now, though if there is high temperature or high concentration of oxygen, the oxygen cannot combine with the RUBP. And by this mechanism, photorespiration is prevented in C4 plants. So due to release of carbon dioxide in bundle sheath cells, the concentration of carbon dioxide goes on increasing. As the process of carbon dioxide fixation occurs in these cells, also more food is prepared through biosynthetic fixation. And as there is no possibility of photorespiration, C4 is more efficient than C3 plants. Now moving on to the another important part, that is CAM pathway. The full form of CAM is granulation acid metabolism. This is another type of carbon fixation pathway that evolved in some plants as an adaptation to arid condition, more specifically of the desert plants. The name granulation acid metabolism comes from the crassula plant, which was the first place that CAM metabolism was discovered and studied. We know that plants must take in carbon dioxide and they must also release waste oxygen, which is the byproduct of the leftover after the carbon atom form has carbon dioxide been incorporated into a sugar. Most plants open the stomata during the day because that is when energy is received from the sun. The energy from the sun is harvested by the chloroplasts and used to make ATP and NADPH in the light reaction. But in plants living in very dry environments, however, dangerous amounts of water can be lost if the stomata are open during the hot, dry days. But during the night, which tends to be much cooler in dry environments, far less water is lost by opening the stomata during the night. This allows the plants to conserve their water by closing their stomata during the hot daytimes. So, what happens during the night? During the night, a plant employing CAM has its stomata open, allowing carbon dioxide to enter and be fixed as organic acids, that is the carbonic acid, by a phosphoenol pyruvate reaction similar to the C4 pathway. And the resulting organic acid, that is the malic acid, are stored in vacuoles for later use, as at that time the Kelvin cycle cannot operate without ATP and NADPH, as the ATP and NADPH are the products of light dependent reaction that do not take place at night. And during the day, the stomata close to conserve water, and the carbon dioxide storing malic acid are released from those vacuoles of the mesophyll cells. An enzyme in the stroma releases the carbon dioxide from the malic acid. Now the carbon dioxide enters the Kelvin cycle so that photosynthesis can take place. Okay? So CAM plants photosynthesis has two phases. One takes place during the day and another occurs during the night. At first, at the night, when they open their stomata, carbon dioxide can enter the cell, and same as the C4 pathway, malic acid is formed, but in this case, the malic acid is stored in the vacuoles until sunrise. And when in the daytime, when the temperature starts to increasing, the stomata are closed. Now malic acid comes outside the vacuoles and is cleaved into carbon dioxide and pyruvate. This carbon dioxide enters the Kelvin cycle, and the pyruvate again regenerate the phosphoenol pyruvate for later use. So what are the advantages of this pathway? The most important benefit of CAM to the plant is the ability to leave most leaf stomata closed during the day. Plants employing CAM are most common in arid environments where water comes at a premium. Being able to keep stomata closed during the hottest and driest part of the day reduces the loss of water through evapotranspiration, allowing such plants to grow in environments that would otherwise be far too dry. And the examples of CAM plants include orchids, cactus, jade plants, etc. Now here you can see the comparisons between C3, C4 and CAM plants. In C3 plants there is only mesophyll cells. In C4 plants there is both mesophyll cells and bundle sheath cells. And in CAM plants, though there is only mesophyll cells, but there are two phases that occurs during day and during night. So let's have a look in the comparison between the C3 pathway, C4 pathway and CAM pathway. In C3 pathway, mesophyll cells of C3 plants are involved. In C4 pathway, the bundle sheath cells and mesophyll cells both are involved. And in CAM pathway, the mesophyll cells of C3 and C4 both are involved. The C3 pathway is observed in all plants carrying out photosynthesis. The C4 plants are mainly the tropical plants, and the CAM plants are those plants of the dry climatic conditions, especially the xerophytes. And obviously in the C3 plants, food respiration is seen in higher rates, but in C4 plants there is no food respiration, but in CAM plants food respiration may occur in the noon time. And the C3 pathway, the first stable compound is the 3-phosphoglyceric acid. In C4 pathway, the first stable compound is the oxaloacetate, and in CAM plant, the first stable compound during daytime is the 3-phosphoglyceric acid, and during nighttime the oxaloacetic acid. Now the numbers of NADPH and ATP required. In C3 pathway, 12 NADPH and 18 ATP are used. In C4 pathway, 12 NADPH and 30 ATPs are used. And in CAM plant, 12 NADPH and 39 ATPs are used. The Kranz anatomy is absent in C3 and CAM plants, but it is present in C4 plants. And the initial carbon dioxide acceptor in C3 pathway is the ribulose 1,5 bisphosphate. In C4 plant, it is phosphoenol pyruvate, and in CAM plants, it is again the phosphoenol pyruvate. Now the examples. The C3 plants are the beans, spinach, sunflower, rice, cotton. The C4 plants are maize, sorghum, sugarcane, and the CAM plants are orchids, cacti, euphorbia. So now it's time for the self-assessment test of today. The link is in the description box. If you have any queries, don't forget to ask me in the comment section, or you can also directly mail to us. In the next Friday, we will learn about the factors affecting photosynthesis, which will be the last part of photosynthesis, and after that I will take a mock test on this chapter. So start your preparation for a better score. And if you are new here, don't forget to subscribe our channel and keep supporting to us. Thanks for watching. See you in the next Friday. Till then, stay tuned with Bioholic.